Hey guys, welcome back to my channel. In this video, I'm going to be continuing the series where I answer biology HL paper 1 questions on camera, such so that you guys can understand what's going on inside my head and also attempt these questions on your own. Seeing as the first video got relatively high support, I thought of continuing this series and attempting another 20 paper 1 past paper questions in IB biology HL. But before we get started, I'd like to remind you that my channel is quite small and it's just getting started, so any amount of help, and whether it's a comment, like, or a subscription is highly appreciated. So just a reminder, if you guys do enjoy my content, please support the channel. It really helps me out. But without further ado, let's get started with these paper 1 questions. As always, if you guys have any comments, any questions with the questions I answer and the methods I use, please comment them down below, and I'll be sure to answer them as fast as I can. So without further ado, let's get started. Glucose is absorbed through protein channels in the plasma membrane of epithelium cells in the small intestine. Which characteristic of glucose prevents it from diffusing through the phospholipid bilayer? So remember that the phospholipid bilayer consists of phospholipids, which are hydrophilic and hydrophobic, which means they are amphipathic. They contain both hydrophobic and hydrophilic regions. The outside heads are hydrophilic, water loving, whereas the inside tails are hydrophobic. So what happens is hydrophilic substance or water loving polar substances have a hard time getting through this layer. This layer is really hard to get through because everything is hydrophobic. So it's probably going to be something without even looking at the answer options. I can tell this is going to be something to do with hydrophilic nature of glucose. So glucose is nonpolar and therefore hydrophobic. That's not true. Its hydrogen bonds link with amino acids in the protein channel. This is not true. Its covalent bonds interact with the phospholipids. That's not true. It is answer option C, which is correct. Next question. In which blood vessel connected to the heart does blood have the lowest carbon dioxide concentration? So if we're thinking about lowest carbon dioxide concentration, we're going to think about when or which part of the heart, which part actually has the highest oxygen concentration, because oxygen concentration and carbon dioxide concentration are going to be inverse of each other. Because oxygen is something that's needed as a nutrient, whereas carbon dioxide is something that's a waste product, right? So if you have blood that's used, it's going to be more like this, where it's low in oxygen and high in carbon dioxide, whereas if you have blood that's fresh and pumped from the heart to the rest of the body, we're going to have high oxygen and low CO2. So where in the body, or which blood vessel connected to the heart has the lowest carbon dioxide concentration and the highest oxygen concentration? So low CO2 means low waste, and because it's low CO2, it's implied that there's high O2. High O2 means that this blood is probably fresh and from the heart. From the lungs to the heart. So which vessel connects the lungs to the heart? Well, that is the pulmonary vein. Veins travel back to the heart, and pulmonary relates to the loop around the lungs where blood is oxygenated. It could be thought that the coronary veins that are non-pulmonary do not have oxygenated blood, so that's straight up eliminated. Pulmonary artery is an exception, which does not have oxygenated, and the vena cava is a normal vein, so that's also not an option. So A is definitely the right answer. Which organ in the human body secretes lipase, amylase, protease? That's just factual recall. It's clearly the pancreas. Which hormone promotes the thickening of the endometrium and also inhibits the hormone that promotes the development of follicle wall in the corpus luteum? So when I'm thinking about the endometrium and its thickening, I already know that it's going to be progesterone or estrogen. So it's neither of these. And if something is inhibiting the hormone that promotes the development of follicle, so this is follicle stimulating hormone. So follicle stimulating hormone is inhibited by this particular hormone. I recall that this is more of a recall question, not an explanation question. I sort of remember it being progesterone, which actually counters or counteracts the development of follicles. So follicle stimulating hormone is what's actually causes follicle development, and progesterone counters that. Like estrogen as well has a bit of a counter effect, but it's more so progesterone. So B is going to be my answer to this question. The graph shows the changes in blood glucose concentration of an untreated patient with type 1 diabetes and a non diabetic person after a meal. So, whenever we get a graph, whether it's paper 1 or paper 2 in a database question, always read the axes. So, here on the x axis, we have the independent variable, which is the hours, and the dependent variable is blood glucose concentration. So when it comes to diabetes and non-diabetic, we've got to remember that type 1 diabetes is to do with it's natural. So it occurs when insulin producing beta cells in the pancreas actually die out. So type 2 diabetes is what you get from eating too much sugar, whereas type 1 is more of a genetic condition. So if there's less insulin, that means it's hard for this blood glucose concentration to go down. Let's see, which statement correctly describes the events in the untreated diabetic patient immediately after the meal was eaten? So this is the untreated diabetic person. We see that the blood glucose concentration decrease is really, really slow, whereas the non-diabetic person has a quick, rapid fall in blood glucose level to that steady level there. And actually, something else to note is that the untreated person has a much higher normal level, normal concentration than the non-diabetic person, almost double about. Alpha cells of the pancreas secrete too little glucagon, so it's not glucagon we're worried about, so we can eliminate those. And alpha cells do not secrete insulin, so we can eliminate that option there. It's going to be D, our answer. Beta cells of the pancreas secrete too little insulin because beta cells have been destroyed by the condition of type 1 diabetes. Type 1 diabetes. A DNA profile was made of one individual in a paternity suit. Locus B was used to distinguish between the individual and other individuals. So this is locus B, it's this strip. The individual has two alleles of the gene at locus B, which are shown below. Okay. Gel electrophoresis was used to separate and visualize the alleles B3 and B5. The gel with two bands of DNA is shown below. Okay, and the question asks us what DNA is in bands 1 and 2. Okay, so band 1 has moved less, so this must be a larger band. So we need to recognize gel electrophoresis as a method, and it's where fragments of DNA are separated according to size, where if it moves further, that means it's a smaller molecule, and if it moves less, it's a larger molecule. So we have smaller molecule, and we have larger molecule. So here we see that for locus B, we have a smaller molecule, which is the B3 molecule, and we have a larger molecule, which is the B5 molecule. So since these bands are far from each other, we can say that these are going to be non-mixes, so it's not going to be a mix one. And band 1 is going to be the larger band, which is going to be band 5. So our answer to this question will be C. Band 5 would be, band 1 would be B5, band 2 would be B3. The diagram shows how pre-mRNA is processed into mature mRNA, which structures are indicated by letters W and X. So if you don't recall, actually eukaryotic cells, right? Eukaryotic cells have what are called introns embedded in DNA exons. So exons are the regions of DNA that are coded for and will actually code for other proteins, right? But the introns are just intervening sections of DNA which don't actually code for anything. So in eukaryotic cells we have exons, but we have introns embedded within them. So because of this, we need to remove the introns when mRNA is made because they're a waste of space. We don't actually translate those mRNA sequences of introns into proteins because that's a waste of the cell's resources. So the structure that's actually used to remove this is actually called the spliceosome, but let's just see what the options are. So W here we see that the X molecule seems to have been cut off, right? So I'm guessing the X molecule is the intron. So this is probably the intron. So let's look at our answer option. I'm guessing this is the intron. So if this is the intron, then the answer option that's correct would be D. D is the only right option. So it could be enticing to think that this darker region is going to be the one that's bad because we've extracted this, and this, it seems like this lighter region is what is going to be transcribed, but no, because it's looped up, we can see that it's going to be thrown out essentially. So we can say D is our answer. Which type of interactions are found in the part of a protein with secondary but not tertiary structure? So secondary structure consists of alpha helices, let me write it properly, alpha helices and beta pleated sheets. And these are due to hydrogen bonding. Disulfide bridges and ionic bonds are a form of tertiary structure. So A is going to be our answer to this question. The diagram shows a mitochondrion, which letter indicates the structure where ATP synthase is located. ATP synthase is located on the electron transport chain, which is a a protein, which is a membrane-bound protein on the cristae, or the folds of the inner mitochondrial membrane. So here we have the outer membrane, we have the inner membrane, and we have the folds, the cristae. So it's actually not going to be located in B, that's the outer membrane. This is the intermembrane space. This is the matrix, which is the inside of the inner membrane, the very inner component of the mitochondria. So C is going to be our answer to this question. This is a cristae part, and electron transport chain is all these benzene folds, and you'll see ATP synthase molecules all the way here that have H plus pumping in towards them, and they have a rotating mechanism by which ADP and a phosphate is fixed together to form ATP. If you guys would like to see me make a video on cellular respiration in future, where mitochondria uses ATP synthase, please leave it down in the comments below and I will get to that soon. Which space has the highest H plus concentration in the chloroplast? So when we talk about H plus concentration, this is about this is about the electron transport chain, because we need to pump out H plus into a particular region so that we create an electrochemical gradient. And when we have an electrochemical gradient, we have a high concentration substance, so high concentration substance that wants to flow down its concentration gradient and into a low concentration area. In this case, we have H plus protons, and when these protons want to move down their electrochemical gradient, they go through this protein called ATP synthase. And through this movement, you get a rotating mechanism which synthesizes ATP. That's the electron transport chain and chemiosmosis. So in the chloroplast itself, we have H plus ions being pumped, I believe, into the thylakoid space. So it's going to be pumped. So if we have a chloroplast, it's going to look something like this. I remember that the ATP itself ends up in the stroma because it needs to be used in the light independent reactions, the Calvin cycle. So H plus is actually pumped into these thylakoids. So it's going to be A. The graph shows the rate of enzymatic activity versus the substrate concentration in the absence or presence of an enzyme inhibitor. So maximum rate of reaction. So we have rate of reaction as the dependent variable, what is measured, and we have substrate concentration as the x-axis, the independent variable, what we're changing, which condition is located or indicated, sorry, by lines y and z. So if we compare, we can see that x is the fastest reaction, and y and z are going to be the slower one and the one that's halfway there. So we can consider x as our default. Y seems to me as competitive inhibition because the reaction, right? Reaction does complete because it ends up at that maximum point where the maximum rate is achieved. But it reaches the maximum at a slower rate, so this suggests to me that there's an inhibitor involved that's competitive. So I'm going to eliminate these two options given with these here. And Z, the other two options say no inhibitor present and non-competitive inhibitor present. So here, since I'm given that non-competitive inhibitor, so no inhibitor present, if there was no inhibitor present, we would have either the default, or in the case that X is not the default, we'll have something even faster, right? Something even faster, but that's not the case. We literally go halfway down. So it's impossible that there's no inhibitor present. So C is our only answer, and it makes sense because a non-competitive inhibitor may not be reversible. So this inhibition process can actually result in the maximum rate never being achieved, which is the case for Z. So C is definitely our answer. Which process and cause are responsible for water uptake by the roots? So the process is going to be osmosis. That's how water moves in. So not simple diffusion or cohesion. This is to do with phloem and the um, pressure flow hypothesis. And solute concentration in the root is greater than in the soil, because this would mean this is hypertonic, and we know that water likes to flow towards hypertonic regions. So B is our answer. Which flower structures are indicated by letters Y and Z? So Z here seems to be a part of the stamen, and this is the anther, and Z is the filament. And here we have the female part, the carpel, so the stigma, the style, and the ovary. So Y seems to be the ovary or ovule, and this one seems to be the filament. I think this one here is the ovary itself, and this is more the Y is more the ovule. So I might go with A as our answer to this question. Next question. Positive phototropisms make a plant grow towards the light. Which plant hormone makes this possible? You should remember it is auxin and cell elongation on the shady side, right? Because if we have so if we have a plant like this, right, and we have the sun shining from this side, if we have growth along this side, what happens is this side grows longer and the plant basically bends this way. If we have growth along this way, it's actually going to bend this way because you're going to have lots of growth in this direction. So it's always going to be the shady side that has the greater cell elongation. So A is going to be our answer. Which gametes can result from the following crossover? So recall that crossing over occurs in prophase 1, and it is the process by which non-sister chromatids exchange alleles. So here we see that the homologous chromosomes have the capital A allele, we have the simple A, capital B, simple B. So what could happen is we can have a capital A and a simple A. We can't have two capital A. Sorry, if crossing over did occur, we can't have two. We can't have two capital A's and capital B's. That would not be possible. So this option is eliminated. See, this is where no crossing over has occurred at all. This option here, it seems possible, but the colors, okay. This one, if we do have exchange, this is a tricky question. Okay, so here we have the exchange occurring. So what's going to happen is we're going to have a B here, a small b, a big B here. We're going to have two big A's, two little A's here. So this one's going to have a big B and a little b like that. So the final outcome should be a mix of B's. So this is not correct. Here, this is not correct because there's allele exchange here. This is not correct because there were two big B's and two little B's. And this one seems to be correct as it's our only other option. And here we see that no exchange of alleles has occurred because the crossing over point, or the, I believe that's called the, I forgot the name for the crossing over point, but it occurs here. So these alleles would be unaffected and they won't mix together. So this will be big A, little b, which is consistent with this answer option D. At first, I didn't see this crossing over point. That's why I was a bit confused. But if you guys don't understand this explanation, please write so in the comments, and I can make a video that's targeted towards crossing over and these types of questions. What can lead to reproductive isolation after just one generation? Polyploidy is not really relevant to this, and reproductive isolation. Well, actually, polyploidy is going to be the answer, right? Because polyploidy would mean that things cannot reproduce the same way. Because if a polyploid individual tried to undergo meiosis, they can't because they generally have an inconsistent number of chromosomes in their haploid number with other members of its species. So polyploidy would be our answer. A secondary immune response occurs when an antigen is encountered on a second occasion due to exposure to an antigen to a pathogen that previously caused infection. Okay, which property of some viruses explains the lack of secondary immune response? Viruses fail to induce a primary response. That's not true. You can have primary responses from a virus. Viruses can have high mutation rate. That's true because if you have mutation, then you can have viruses that are basically new versions of themselves, such that the previous antibodies you produced on the first exposure don't actually affect them anymore. So B seems to me as the correct answer. Which hormone is inhibited during pregnancy in order to prevent contractions? The contractions of the uterus are from oxytocin. That's a positive feedback loop. So that's just basic recall, and the A is our. The table shows solute concentrations in normal blood plasma and the fluid in one section of the nephron. So we have the solutes, normal blood plasma, plasma level, okay, fluid inside nephron. Okay, so when it comes to glucose, in which section of the nephron would you expect to find these concentrations? So firstly, we see that the chloride ions are the same, the urea are the same, and proteins are they're quite much lower. So it looks to me as proteins are the only components that have been filtered out or filtered into the nephron. And it says the answer options were removed for copyright reasons. So great. We don't know the answer to this question, but I'll make a video on the nephron and how to compare these blood plasma concentrations if you guys would find that beneficial. So if you guys think that this question would have tripped you up, so how you would answer this is compare the plasma concentrations and find out which part of the region it's not reached at, or which part of the region has this type of blood plasma and the fluid inside the nephron because of the lack of filtration in these ones and the filtration that's occurred in this one. So you got to use this information to determine the location. So if you guys want me to cover that in more detail, please write so in the comments down below, and I'll make sure to make a video on this topic to help you guys. So let's do this last question. The diagram shows the exchange processes that take place in the placenta between the maternal and fetal blood, which process requires endocytosis. So maternal blood would be providing for the fetal blood. So water to the placenta to the fetal blood does not seem to me like something. Okay, that's not even an option, anyways. And oxygen to the fetal blood, that would be from diffusion, so you don't need endocytosis. CO2 again, that's a gas. Urea. Immunoglobulins are quite tiny. They're proteins, so they're not going to require. Actually, this is interesting because immunoglobulins and glucose are our only other two options. And immunoglobulins, considering that they are proteins, they should be proteins are thousands of molecules, thousands of amino acids in length, right? So a glucose molecule is so tiny compared to that. This will be millions of times bigger than the glucose molecule. So our logical approach to think what would require endocytosis, meaning it's a big molecule that cannot get through normal diffusion, I would have to say D is going to be the best answer. I'm not sure. I don't think I've even encountered this question in the syllabus or this information in the syllabus. It's a weird question, but I think you're going to deduce from the sizes which one is more likely to follow endocytosis. Alrighty, guys, I have checked the mark scheme and it turns out we got 19 out of 20 of the questions right. So it's a really good score considering we did quite hard high level questions towards the end there, especially. And that's really great work. And if you guys were following through and understood everything, that's big thumbs up for you. And if you guys didn't, just comment down below anything you misunderstood and I'll be able to address that in future. This was the question we got wrong. So the ovary, I accidentally named this part the ovary. Yeah, wait, no. I've written it properly. I've somehow answered it wrong. So D was the correct answer. So Z was the filament. And this structure, so pretty much the ovule is this thing, the actual female gamete, and the ovary is the structure that contains this. So if it's pointing here to the overall structure itself, this is the ovary. If it was pointing here, then that would be the ovule. Other than that, guys, hopefully you enjoyed this video and found it helpful in your revision to gain a 7 in IB Biology HL. If you guys did so, please don't forget to support the channel, like, subscribe, and comment down below anything you guys want. It really helps out the channel because this channel is quite small. Any small help is highly appreciated. Other than that, guys, hopefully you guys can find my other videos helpful as well and learn some more. And see you guys in a future video. Peace out.